While everybody's settling down in the coffees, I think it's time to get started. We're a few minutes late. Anyway, it's summertime, we can take it a little easy. So, first of all, I'd like to welcome you all for the PHRN seminar. As you know, we have once a month, the seminar series going on a regular basis. And we also accommodate the guest speakers, the guests who are visiting Guelph, so that, we can listen to them. And then they're going to share their data and the research with us, which is pretty exciting. So in this context, our guest speaker is Dr. You-Chan Bae, who is a senior researcher at the Plant and Animal Quarantine Agency, Republic of Korea. And Dr. Leo Suspa is hosting him, and he's here for the entire week visiting the pathobiology as well as the animal health laboratory. And I would like to pass on Leo for introducing him, since he's the best person.
Okay. Thanks everyone for coming. It is my pleasure to introduce Dr. You-Chan Bae as our speaker today. Dr. Bae, I'm going to give you a little bit of background about his history. Dr. Bae received his DVM in 1990s from the College of Veterinary Medicine in Chennai. If you pronounce that right. And after his DVM, he completed a Master of Science studying Haemophilus parasuris. And after that, in 2005, he completed a PhD studying bovine papillomavirus. At the same time, while he was conducting his graduate studies, he was also working as a diagnostic pathologist at the Animal and Plant Quarantine Agency. And in the last few years, he became more interested about avian medicine and avian pathology. And so in the past few years, he started to work in the avian division as a senior researcher and pathologist. His interest is the pathogenesis of the Enterococcus cecorum and the high pathogen influenza, and of course, pathology diagnostics of poultry species. Here at the OBC is planning to establish some connection with other faculty in the pathobiology and also with other diagnosticians, both pathologists, microbiologists, and biologists at the Animal Health Laboratory. And with this, I will give Dr. You-Chan Bae the floor.
Thank you, Leo. I'm from Korea. In this morning, I will talk about pathological changes in wild birds, chicken, dog infected with HPAI, H5N8 strain in Korea. We have 5 outbreaks of HPAI. This slide shows the first case, 2003 years. You can see the approximately 100% of mortality within 5 days after onset of clinical signs. As you know, HPAI is very devastating and highly pathogenic disease. Avian influenza is a contagious disease, birds, animals, and humans. The virus has been originated from free-flying aquatic birds, especially wild ducks, mallards, bicartil, and etc. So we are thinking about the genetic reservoirs of all AI virus is wild ducks. Domestic poultry flocks are especially vulnerable to this disease, especially duck, chicken, turkey, and quails. In Korea, we have two major poultry, chicken and ducks. AI is divided into 2 forms, low and highly pathogenic. This slide shows about pathogenesis of AI, HPAI. As you know, AI virus infects the chicken or duck by oral or nasal routes. And then they replicate in respiratory epithelial cells. And then they move to the vascular endothelial cells in respiratory, in capillary endothelial cells in respiratory system. And then they move to the whole organs, parenchyma, brain, and skin by blood and lymph. And then they induce devastating damage in avian species. First one is direct virus replication in whole tissues, or they produce cellular mediators such as cytokines, or they induce damage by ischemia from vascular thrombosis. And last one is they induce cardiovascular collapse from DIC. Therefore, the virus induces highly pathogenic lesions. As I said to you, we have 5 outbreaks of HPAI. First one is 2003, and then 2006 and 7, and 2008, and 2010 and 11, and the last one is 2014 to 2016. For previous 4 cases was caused by H5N1, and clade is different. But last one is caused by H5N8. The clade is 2.3.4.4. This is cost for eradication. The last case is more than 238 million. So it's very devastating. I will introduce the last one, 2014 case. January 2014, we detect H5N8 in breeder dogs. This farm is located in Gochang. Gochang is located in southern part of South Korea. The breeder dog shows decrease of egg production more than 50%, and increased death and depression. We detect H5N1 within 2 days. After that, we detect the dead baikal teals in Dongnim reservoir. This reservoir is very near from index farm. Index farm means these farms. In Dongnim reservoir, we find out 100 of dead baikal teals. So we find that this poultry case is closely linked with wild bird. So we think that the wild bird is first one, and then the virus spread to the poultry. This slide shows breeder dogs' growth lesion. You can see the severe ovarian rupture and hemorrhage, and then swelling, hepatic swelling and hemorrhage also. This slide shows the results of necropsy of more than 700 wild birds collected January to June 2014. We classified into 2 groups. One is pathogenic case. The other one is non-pathogenic case. Pathogenic case is at 18%. And HPAI virus was isolated from 100 67. And bacterial infection is 29. Parasite infection is 9 cases. Among the non-pathogenic case, organophosphate phosphate poisoning is more than 220. And gunshot is 12. Trauma is more than 100 cases. This slide shows gross lesion of bical teal. You can see that torticollis by neurological sign. In histology, we find out severe perivascular coughing and gliosis in this bical tear. You can find out severe hemorrhage and necrosis in myocardium, and severe hemorrhage and edema in lung. And the most important lesion is coagulative necrosis in pancreas. This pancreatic lesion is very prominent lesion in AI. This slide shows the wild birds which was infected by H5N8. The most of wild bird is bical teal, more than 120. And bean goose is 9 cases, and mallard is 1 case, and etc. This slide shows histopathological and immunohistochemical results of AI virus antigen using 11 wild birds. Most of case is bical teal at 8 cases, bean goose 2 cases, upper swan 1 case. The most important lesion was detected in brain, kidney, and pancreas and liver. This slide shows histological lesion and immunohistochemistry of bical teals. You can see the coagulative necrosis of exocrine gland in pancreas, HNE, and this is immunohistochemical staining using AI common antigen. You can see the numerous AI virus in necrotic area. This slide shows the kidney. You can see the tubular necrosis, and then you can detect the AI antigen in necrotic tubular epithelial cells. This slide shows histological lesion and immunohistochemistry of bean goose. This is a pancreatic necrosis, and you can find out the AI antigen in pancreatic necrotic area. This is a heart, myocardial necrosis and inflammation, and you can see the AI antigen in necrotic myofibers. And this slide shows the peri-Purkinje cell, Purkinje area vacuolation, and you can find out AA antigen in necrotic pancreatic cells, necrotic Purkinje cells. This is a cerebrum. You can see the severe perivascular cupping and gliosis. And you can see the AA antigen in neurons or glial cells. This slide shows the layer HPAI cyanosis in water. This slide shows the skin of HPAI. You can see the neurological signs and cyanosis. As a differential diagnosis, I suggest colibacillosis septic form. This case is a broiler 29 days. You can find out cyanosis in face and abdomen and other skins. When I do necropsy, the hepatic swelling and pancreas and splenic swelling and severe swelling of kidney, and I isolate the pure culture of E. coli from liver. So I think that we have to think about colibacillosis as a differential diagnosis. This slide shows hemorrhage in subcutis and trachea, HPAI case in chicken. This is a normal lung. These two pictures is HPAI in chicken. You can see the severe hemorrhage and edema. Yes, severe hemorrhage in both sides of lung. HPI layer, you can see the epicardial hemorrhage. Duct HPAI, hemorrhage and necrosis in myocardium. And this is an advanced lesion. You can see the severe necrosis in myocardium. This slide shows Limerella anaphysativa in mid-duct as a differential diagnosis. You can see the fibrinous epicarditis and hemorrhage and fibrinous perihepatitis. In Korea, we think about the Limerella is important disease as a differential diagnosis of HPAI. This is a chicken HPAI. You can see the severe hepatic rupture and hemorrhage. This slide shows the differential diagnosis of proventriculus HPAI, Newcastle, and IBD. So it's not easy by gross lesion. So we have to do virus isolation for differential diagnosis. And this is very important gross lesion for HPAI. You can see the whites or grayish whites, necrotic foci in pancreas. Yeah. It is a very typical and important lesion in HPAI. This one is also HPAI chicken, splenic swelling and ovarian hemorrhage. This is a duct of spleen. You can see the severe swelling and necrosis. HPI chicken, severe hemorrhage in skeletal muscle. HPI duck, you can see the meningeal congestion and hemorrhage. Okay, this slide shows isolation of H5N8 virus in poultry and wild birds. We isolate 391 from poultry and 58 from wild birds. We have 3 waves of HPA outbreak from 2014 2015. You can see this is a winter season in Korea. So in winter season, the numerous virus was isolated. This is a western, and this is a southern. the most of cases from western and southern area. This slide shows the affected species in wild birds. Most of cases from Baikal teal, and then mallard and bin goose and white-fronted. And we also H5 antibody from mallards and Baikal teal and spot-billed ducks. This slide shows affected species in poultry. Among 391 cases, duck is 75%. So the duck is very important species for HPAI in Korea. And chicken is 22%, others is 2%. Among duck, meat duck is most important source, more than 230 cases. And breeder duck is 59 cases. And for chicken, layer is most important species, and Korean native and breeder duck, breeder chicken. This slide shows phylogenetic analysis of H5 gene. This is a Korean strain. The blue one is a Chinese strain. So we found that the Korean H5N8 strain is closely related to the Chinese strain. This slide shows that points. These three virus is from Eastern China. The China duck, China environment. These two viruses is a Korean strain in 2014. You can see the group A virus is from China, and also group B virus is from China strain. Okay, thank you.
Thanks, Dr. Bae. Now we'll open for questions.
Thank you. My question is about the susceptibility of the white birds to this HPAI viruses. Is there a difference between ducks and any other water inflammatory wild birds or not? In the meantime, is there any difference in the susceptibility of wild ducks and wild domestic ducks?
Good points. Well, maybe mallard. Okay. I think that the chicken and duck and Baikal teal and bin goose is a same lesion, but mallard is very resistant to HPAI virus, so they did not show any lesion. Yeah. Yeah.
Okay, thank you for the presentation. So you were talking about the pathological signs and lesions for the H5N8 virus. Would that be different for the H5N1 virus? Or based on postmortem, can you differentiate which virus you are dealing with?
Okay. It's not easy to tell about the growth lesion of natural case, but when we do in fact, experimentally, the lesion caused by H5N8 is less pathogenic than H5N1. Yeah.
So there's the lesion, so it's actually less severe.
Yeah, less severe.
clinical signs.
Yeah. Clinical signs and lesion is less severe. Yeah.
So what is the situation now in Korea? Because
Now?
yes, you have.
No. It's all gone.
Oh, is it? Okay. No, because you said ongoing.
Ongoing. Oh, yeah. Mm-hmm.
Thank you.
I have a question about the China isolate. You're talking about the Korea isolate related to China. What about, do you know the pathogenicity of China isolate? It's very highly pathogenicity. You know, the last slide you showed. Do you know that China isolate? These isolate are highly pathogenicity in East China. You showed 3
Oh, yeah.
isolates
Isolate.
from China, Jiangsu and
Yeah.
they are highly pathogenic
Okay.
or lower.
No, not.
Yeah,
This
that's-
is environment, so I cannot.
Yeah, the middle one, the Jiangsu, that's same, it's 5N8.
I
That's
think, yeah, this one may be highly pathogenic,
okay.
this one. And this one is also highly pathogenic, I think. Yeah.
So you're asking if the Chinese isolate or strain is more pathogenic than the Korean?
Yeah, I'm just curious, maybe during the time the originally in China, let's say 5N8 or lower pathogenicity doesn't cause disease. Maybe after a while, yeah, something happened. So in Korea, that H5N8 cause the severe disease. I don't know.
Yeah.
Can you show us the slide that has the 3 waves?
Mhm. Yeah.
I'm just wondering if you could maybe explain what was sort of happening in the country at that time in terms of government or industry interventions, or was there quarantine or
Yeah.
something
Yeah.
affect
Okay. Culling
culling?
and quarantine and disinfection. It's all measures was done, but it's not easy to eradicate. So, yeah, it's not easy. Yeah.
I've got a question. Do you guys in your laboratory conduct standard pathogenicity indexes, like IDPI?
Yeah.
Do you guys explore these isolates? What was the IDPI?
I don't remember that because the other section was done, but yeah, that's a highly pathogenic index. Yeah.
And also, I have another question. When you say about EPI differential diagnosis, I guess in morphology, sometimes you have, when you have CPC, maybe so those kind of things. But what about presentation? I mean, I assume that the mortality rate H5 and 8 would be much higher, right?
Exactly.
For such short time. That's your experience?
Yeah. Thank you. The HPI mortality is more higher than colibacillosis. Yeah. Exactly. Thank you.
Okay, if there are no more questions, thank you very much, Dr. Bae, for the brief and sweet presentation. I think it was the quickest ever. And please join me in thanking you.
Thank you. Thank you, Roger.
We still have some coffee and tidbits left, and I also want to say, like, in our series, seminar series, so this is going to be the last one in this particular season. So we're going to take a break in June through August, and we'll come back in September. And thanks for your help and support. Thank you.